E. coli, the bacteria in your intestines, has become the ultimate creator. From fuels to rubber to insulin to even cancer research, E. coli has become very useful to us. And while I'm not a biologist, I am a chemist, so let's get into chemistry and try to break down why this is. To the lab! So this cute guy over here only has 4,400 genes, which is relatively small to the 30,000 genes that humans have. Since this is only about 15% of the human genome, scientists love to study him. I mean, isn't he so cute? Whoa, 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 not so fast. You see, scientists treat E. coli more as slaves rather than cute pets. Unlike human cells, bacteria have something called plasmid, which has a ring of DNA separate from the chromosomal DNA. Essentially, this is like power-ups which bacteria use to help fend off antibiotics and helps them survive in harsher environments. What scientists love about it is that it can duplicate itself many times without E. coli reproducing. And then when E. coli does reproduce, it has identical plasmids in both the parent and the child cells. So is anyone catching on what these mad scientists are doing with E. coli to help bend it to their will? Well, let's look at a specific example, insulin. Insulin for most people is naturally produced inside our cells, but if you have diabetes, this typically tends to be harder for the body to produce, if at all. So if you're diabetic a couple decades ago, the only way you were able to get insulin was through pigs, and this tended to be super expensive and hard to do. Scientists were looking for a different way outside of these pigs that they can produce insulin for a lot cheaper. And you know what they used? E. coli. So how do we get this E. coli to produce insulin? What scientists do is add an enzyme or a specifically designed molecule which breaks that DNA ring, the plasmid, and they can separate an unwanted portion of the E. coli's DNA ring and insert a wanted portion. And so what do? We take the DNA in humans that produce insulin, and then we take that DNA and insert it back into the plasmid ring. And now that this E. coli has DNA inside of it that tells it how to produce insulin, E. coli now thinks it needs to produce insulin and able to survive. But now you might be thinking,
E. coli is really bad for us and it'll make us really sick. How do we get over that fact?
So what they do is they just put it in a big batch, and after the E. coli produces a ton of insulin, they just murder all of the E. coli cells. So now that you know this, you can kind of understand that scientists are just using this same method and putting in new DNA strands into E. coli to force it to produce what we want. So now do you believe me when I say that scientists use this to make rubber and diesel fuel? Well, what about if Peter Parker used this to produce his spider web? Interesting.